right, Dr. Momo Vuyisich, thanks for taking the time to be on the Bioinformatics Lab podcast. Turns out for the past 5 years, you and I were neighbors, but we didn't really cross paths till I saw you make a post on ube cappuccinos. So I'd like to start there, man. In what way does purple coffee impact your microbiome?
I don't think, well, so ube that I bought, which is powdered, I don't actually know what kind of processing they do, but sweet potatoes have some really, really good prebiotic fiber, right? And I think that sweet potatoes are likely one of the reasons that these, East Asian populations have such long health span because they're feeding their microbiome really well with sweet potatoes. And so Okinawa is obviously famous for sweet potatoes. I prefer these Filipino, sweet potatoes. Ube is just a little bit more flavorful, and the color is incredible. So we've been eating ube for all kinds of reasons, but this ubecino is like a new thing. I just found out that from a friend on Facebook that they make cappuccino with ube in some New York coffee shop. And I was like, wow, why didn't I think about that? So went to Amazon, bought the powder. 2 days later, I'm fine tuning my recipe and now it's already done. It's like that simple.
I love that, man. Ube's in vogue. And I'm from Filipino heritage, so I grew up with like ube all around, sweet potatoes, as you mentioned, part of our cuisine, especially on the dessert side. And I don't know if you knew this, man, but in downtown Bellevue, there's a cafe called Cafe Holics. They've got an ube cappuccino.
I did not know that, but I generally don't go to cafes in Bellevue. They're a little bit above my pay grade,
Fair. Well
if you
said.
know what I mean.
I think anyone in the Seattle area knows exactly what you mean, man. But in that, obviously, we cross paths because you're out there in Washington. You got a lab out there, Biome. I can't believe I was out there for so long. I didn't know much about what Biome was doing. I'm in the pathogen genomics space. In my background is in microbial bioinformatics, so I was always tangential to what was happening in the microbiome space a little bit. But can give a general overview. You're tackling a problem in a way most aren't. And maybe we can start at metatranscriptomics versus metagenomics and why your team chose the route you chose.
Yeah, so that's a long topic, of course, but let's talk about a little bit about human evolution first.
You
So
just made a big topic bigger.
but really the context is very important. Why metatranscriptomics over metagenomics? So the context is human evolution, and we have evolved, our physiology has evolved to depend on biochemical reactions. And I would estimate that there are about 100,000 biochemical reactions powering our body. And that starts with our muscles moving our mouth to ingest food, and then digestion of food, transportation of food to all the cells, mitochondrial energy generation, and then neurotransmitters, and then central nervous system and liver and kidney detoxifying our body. All of these biochemical reactions, immune system biochemical reactions, 100,000 roughly. And also my estimate is that about half of them are run by our own human genes and half by the microbial genes, roughly. This is rough order of magnitude. I don't really want anyone to quote me on the exact numbers, but roughly. And we have co-evolved with these microbes where there's a chemical messaging going on at all times in order to maintain our homeostasis. And that's the key, that it's chemical messaging, and it only depends on the actual chemical. As we were evolving, our immune system has never learned what Methanobrevibacter smithii means or what Faecalibacterium prausnitzii means. And still to this day, my immune system and your immune system does not understand the concept of Faecalibacterium prausnitzii. Like, you cannot tell your immune system, oh, my result says that I have Faecalibacterium prausnitzii in my gut, so you should be happy. You should get plenty of butyrate, right? Our immune system doesn't understand that concept. That's a human-made construct. The chemical construct that we have evolved with is that there's a chemical called butyrate that may or may not be produced by Faecalibacterium prausnitzii that signals the immune system to do certain things. And the example that I love the most is, for decades, we have known that if you vaccinate 100 people, not all of them will successfully vaccinate. right? There's vaccine efficacy, and it's anywhere from 50 to 95%, but it's almost never 100%. So there's always some people who don't respond and to vaccination, and they don't develop immunity. And that was unknown as to why that was until pretty recently, like 2023. It turns out that butyrate that is made in situ in the colon, when it diffuses into the bloodstream, it's a key component that basically turns on a signal transduction pathway in cytotoxic CD8 plus T cells and stimulates the pathway that transforms those cells into memory T cells. And it's those memory T cells that provide us with the lasting immunity. If butyrate is not produced in the late stages of a disease or a few days after the vaccine, then those cytotoxic reactive CD8 plus T cells will simply die off and you don't get a memory. Okay. And this mechanism was I think I understood this mechanism probably as early as maybe 15 years ago, in that UC Davis scientists studied vaccine responses in Bangladesh, and they realized that there was a microorganism, bifidobacteria, that were very strongly predictive of vaccine efficacy. And then they isolated Bifidobacterium longum strain infantis. And in fact, I think now a lot of people just call it Bifidobacterium infantis as its own species. We don't have to argue about that sort of taxonomy level. But now, mechanistically, it's been shown that it's butyrate, the product of that microorganism, that that causes this cascade of reactions. And so here, the reason why I like this example is that there's a requirement for specific micronutrients to be consumed that match the microbiome that you have. And then that substrate has to become preferred substrate for your microbiome in order to basically feed on that prebiotic, produce butyrate, and then stimulate your immune system memory.
Okay, there's a ton to unpack there. A couple threads I want to pull on there. So if I can kind of summarize the position you're taking there when you're looking at microbiome interactions, the premise you're standing on there is gene expression matters more than gene content. Gene content is almost, that's what's there, but that's a proxy for what's actually being expressed. Your aim is what's actually being expressed to give us that insight into what's actually happening mechanistically.
Yeah, that's exactly the next point that we need to focus on, and that's a major takeaway. In our data, when we do taxonomic classification of a stool sample, the average number of genes encoded by a person, one of our customers, if we take 100 random customers and look at the taxonomic classification and then just enumerate all the genes, an average gut microbiome encodes 4.5 million genes. But of those 4.5 million genes, less than 100,000 are actually expressed to any appreciable level. And we set that threshold at 1 part per million, or 1 CPM, right? And then not only do we now know which 2% of the genes are actually expressed, but we can actually quantify the level of expression of those genes, because the level of expression also matters for connecting microbial activities to different phenotypes. And really, that is the core of this discussion, that you cannot just say, well, my microbiome has a gene potential of 4.5 million genes. My microbiome does all these things. You have zero information as to what your microbiome is actually doing. Zero, if you're doing any kind of a DNA analysis. You can only say that your microbiome has the potential to perform those biochemical reactions, and that's it.
Okay, yeah, okay. I hear you exactly what you're saying, but so is the argument there that it's a poor proxy? So if you saw something like you mentioned, the bifidobacterium, if you identified it in a microbiome sample at a certain prevalence, that's still not enough to say that you're going to mechanistically see that phenotypic expression that you described.
That's correct, because the microbes will express only the genes that are the most beneficial for their proliferation. Yeah. And so there's a lot of direct feeding on micronutrients, but there's a lot of cross feeding, and it's a community. And you don't necessarily, no species can say, well, I'm just going to take whatever I want, right? There's a lot of community and synergy. And so, yes, exactly. You cannot say that just because you have the genes that those biochemical functions will be performed.
Hmm.
You have to measure them. And obviously, the best way to measure them would be to look at the metabolic output using untargeted metabolomics.
Uh,
But we are simply not there yet technologically. That's simply not a feasible, a practical analytical method today for massive direct to consumer analysis. It's just not. Maybe in 10 years it will be, maybe in 30 years it will be, but I think that RNA is going to help us solve the vast majority of these problems without getting to metabolomic analysis.
all right. I was just going to ask that question. If you could not just walk that logic all the way down the central dogma, if you're saying not DNA is too much of a proxy, then that common argument would be so is RNA, because not all those expressed genes turn to proteins. Not all those proteins are necessarily functional. The metabolites are what matter. But it sounds like your contention there is there's too much noise in that data to actually be actionable.
Well, it's just, direct to consumer metabolomic tests are not practical yet. They're too expensive. And we don't know the vast majority of metabolites, right? The vast majority of, if you do untargeted metabolomics these days in blood and stool, you may know 10% of them. I'm not even sure it's there yet. And so there's a huge dark space. And then if you identify metabolites, let's say you identify metabolites that are associated with some phenotype of your interest, you still don't know exactly which microbes are producing those metabolites necessarily, whereas RNA sort of ties that composition to the metabolic output, to the phenotype. And so I think RNA is the single most powerful omics technology for systems biology, understanding the interactions between nutritional ingredients, microbiome functions, and then human physiology.
That's a fascinating way to look at it, right? Yeah, because you're right in the middle. And I think actually maybe more important to what you described, the technology is available. We have sequencing, we have RNA-seq, you can do it at super high throughput, whereas you
Well,
haven't seen that necessarily leaps in technology at the protein level or maybe metabolite level either.
exactly. No, but I mean, that's exactly. So in 2010, when I envisioned this whole platform that we're running today, I realized I can probably make metatranscriptomics work. I cannot get proteomics or metabolomics work. Plus, I really like that RNA sequencing gives you the full taxonomic classification profile, which is important. And so it sort of crosses that world from composition to function. It's quantitative. At that time in 2010, it was very expensive. It's still very expensive. But over a period of 6 years, my teams solved sort of fundamental problems with metatranscriptomics in terms of accuracy, reproducibility, scalability, cost, automation, all these things for 6 years. We developed it, we patented it, and then in 2016, I spun out Viome with Naveen Jain, and we licensed that technology from Los Alamos National Lab.
And that's another aspect to this too, because I'm so curious on the business side of the Viome decisions, because exactly what you said, in my eyes, when I think about metagenomics or even metatranscriptomics, I'm still thinking like north of 1,000 for an assay to get proper coverage that you need to actually properly profile. But then I'm
Yeah,
seeing on Viome site where it's like a couple $100 and I got a whole
amazing,
profile.
amazing,
So
right?
are you able to speak to the mechanisms that allow that kind of economic price point?
Well, yeah, it's 6 years of 7 days a week work to
That's
get to
all it
that point.
took.
So that's all it took, right? Well, first of all, I have to thank the US government for funding the vast majority of that work. I also have to thank all the program managers that believed in me and that didn't require all those publications, which would have just distracted me from accomplishing this goal. And then, so there were many steps, but basically, yeah, you're right. So I'll just give you one example. If you want to do ribosomal RNA depletion, just that, and you want to buy a kit
Yeah.
off the web, right? The kit costs anywhere between $50 and $100 per sample
Yeah.
for ribosomal depletion, right? Our method does ribosomal depletion and more, and it does it for $1. Okay, and it's fully automated,
Yeah.
and it's better performing than any commercial product that's available. And so that's one slice of the whole thing. The second slice of the whole thing is, well, there's many slices. So let's start from the beginning. Problem number one is RNA degrades fairly quickly after sample collection.
Notoriously
And
so.
I worked all over the world while I was a government employee, and it took sometimes a full year. It wasn't the norm, but it took some time, months or a year, to get a sample from Kenya or Uganda, because you had to get USDA permits and CDC permits, right? And so immediately I knew that you can't scale, you can't have a global business if it takes you 4 months to import a sample. So you had to use some kind of a preservative that can preserve RNA, but that can also inactivate all pathogens. And so we worked that out. We basically worked out a formulation that instantly kills all pathogens, viruses, bacteria, and fungi at the time of collection, and preserve RNA for a month at room temperature. So that was a key. That was a key, the movement. The second is, if you're doing any kind of a metagenomic analysis, and especially 16S, but even just pure metagenomics, you're going to find very soon that your water controls have their own unique microbiome. Every time you order new reagents or new plasticware, whoa, now all of a sudden your background changes, right? And it goes up and down. And so what we did for several years is we worked with OEMs to develop reagents just for our own process. So today, none of our reagents are off the shelf. Nothing. No, not the water, not the buffer, not the enzymes, nothing. It's all custom made for us, such that the background is very consistent and very low. And so that's a multi-year project right there. Next thing is that cell lysis and RNA extraction. Cell lysis specifically is the most overlooked, most underrated, and most biased part of the entire method in my mind, that people simply brush off and say, well, QIAGEN has figured all this out. They've been in business for 40 years. Their lysis must work. Until you say to yourself, you know what? I'm going to challenge my test, and I'm going to get my test from a research grade to a clinical grade. Let's do a full CLIA validation and see, does that lysis actually pass that? And it turns out none of the MoBio or QIAGEN or Promega or any other kit on the planet could produce reproducible results. The cell lysis was biased. And every time you analyze the exact same sample, you would get different results from technical replicates. Different enough to where if you're trying to do machine learning from different samples and your noise is huge, machines are going to pick up on those biases, not on actual signals. So it took us some number of years to develop our own proprietary cell lysis, mechanical and chemical lysis, to where it would pass very tough criteria for a clinical validation. And so today, we don't buy any kits for cell lysis or extraction. It's all our own reagents. And that was a critical component that, like I said, most academics and most companies, they simply say, buy it from QIAGEN. Like, how many publications does QIAGEN have? Million, right? Then we go into this depletion, which we kind of discussed, right? 95 to 98.
It's like a hybridization approach.
Well, there are many ways that people do it. We do subtractive hybridization. So very simple. We design our own probes, DNA probes that are biotinylated. We capture the RNA sequences we don't want in the sample, and we fish them out with magnetic beads. Very simple. Hybridize, cool, and capture with streptavidin beads. Very simple process, but the trick is in the design of the probes to have the right specificity for the sequences you want and not affect the results. And so that's not a trivial process. You have to adjust buffer conditions, temperature, probe length, probe specificity, lots and lots of different things. So we have worked out that depletion process to where in stool and blood, we have more than 50x improvement in terms of getting to the sequences that we want, which are non-ribosomal, right? And so that makes the whole process downstream at least 50 times more efficient. So that's very nice. And then comes probably the single biggest factor in our success in terms of machine learning is barcode hopping. Are you familiar with barcode hopping?
Oh, absolutely. I was going to ask about exactly this. How do you do contamination controls in this conversation, especially with such messy data, not having... Yep.
So interesting thing about barcode hopping is that everyone knows about it, but very few people do anything about it, right? My story on that is that back in 2012, we brought some blood samples from children in Kenya who had malaria, and on one flow cell, whatever sequencer we had in 2012, some old Illumina model, On one flow cell, we sequenced 25 blood samples, and one of those was from a Kenyan child with malaria. The other 24 were from people in New Mexico. And the bioinformatics comes back with taxonomic classification, and it says Plasmodium falciparum is present in all 25 samples. And I say, you got it wrong, guys. That's not possible. These 24 people have not been to Africa or any other malaria exposed region. There's no way that they can have it. And they're like, no, we're getting actual Plasmodium falciparum reads. You can confirm that by blast. They're absolutely super specific. There's no doubt. And so we then realized that this barcode hopping is producing about 1% noise in every single sample. 1% of reads in one sample come from samples on that flow cell and not from the original sample. And so now you have 1% noise. And as you know, with taxonomic classification or gene expression, the vast majority of features that you're quantifying have a relative abundance that's less than 1%. How are the machines going to now pick up the signal when your gene expression is in the 10 parts per million and your noise is 1%? That's not going to happen. And to me, that was a showstopper. To all other people that I spoke with, they say, well, that's the limitation of the Illumina technology. What are you going to do about it? You just live with it and you just do the best you can. I'm like, no, you can't live with it, right? So it took us 2 or 3 years to work this out and identify all the sources of barcode hopping. And in 2015, we were able to patent that process, and we are now the sole patent holder for that. So that was pretty exciting. So now we've moved that noise floor down to 1 part per million. So we have sample-to-sample crosstalk at 1 part per million or below, which now means that the vast majority of features that we measure, whether they're taxonomic or gene expression, are above that noise floor. And so that is, I would say, one of the main reasons that our machine learning works so well, in that we have good signal to noise.
I think the through line I'm hearing at each one of those spaces of innovation is this concept of first principles thinking. Let me get back to what's actually happening at the biochemical level. Let me not assume that the large manufacturers have figured it out and they've tested the limit. Let me tease this apart with my lab. Let me take 6 years in my dungeon and I'll figure this out. And then I'll tell you if it's possible. And it sounds like at almost every step in this, from extraction to prep, library prep, you've reinvented everything except for sequencing at this point.
That's exactly right. And our sequencing uses custom sequencing primers. It doesn't use Illumina sequencing.
Oh, fascinating. Yeah.
So even that is slightly different for various reasons. It has to do
You're
with
just using their camera.
We're using their camera there. Exactly. So yeah, it took 6 years to work all this out to get really accurate data, really reproducible data on any human clinical sample, and then optimize the process so that today, when we put on saliva, blood, or stool samples on our robots, they're pulling from the exact same method. They don't actually know what sample they're processing. The method is identical. And that's a massive, massive, massive problem to solve. If, running a clinical laboratory is extraordinarily complex in terms of preparation of reagents, qualification of reagents, storage of reagents, method writing, method improvement, method quality control. If we had 3 methods for stool, blood, and saliva, I mean, it just might be cost prohibitive to run a clinical lab for that. So one of the major advantages of our method is that it's universal. You can go to FFPE, you can go to soil, you can go to CSF, urine, you can go to skin. It doesn't matter. The method is universal. So that is one of the big reasons it took 6 years as well to develop one method that is a true metatranscriptomic method for anything.
It sounds like, too, now we've got this 6 years plus whatever other time prior to, understanding the science behind everything and building the resilience to push these things through. Undeniable methods development to bring RNA-seq, again, to a not only a optimal scientific clarity there, but also economically, it's something now that there's a market strategy. But I think, so if we're starting there, that's our base point. Okay, you know what? Momo now has the capability. He knows how to get expression profiles from these complex samples, be it your gut, I think you mentioned oral, and also, was it blood as well?
Yeah, whole blood, stool, and saliva for as direct-to-consumer products. We, of course, for research, we do many other sample types.
And so if we're starting at that point, okay, for sure, you're getting the gene expression. You're not just looking at taxonomic breakdown. You're looking at functional based on what's actually being expressed. now hop us into that conversation. How does that become applied? Because the other side of that is I need a context for what the heck I'm looking at to be able to intervene medically, nutritionally, etc.
Yeah,
How do you translate these gene expression
yeah,
profiles to something that, okay, now I should eat kale instead of spinach?
yeah. Okay, we will absolutely get to that, but I would like to stick a little bit with what we just
Please.
discussed because I think it may be of interest to some people. So I hear a lot of academics asking me, how did you decide to leave academia and start a company? Isn't that like super risky? How did you learn that stuff? How was your first day? Were you scared? All that stuff, right? So I give advice and I give whole seminars on this topic, and I just want to spend a few minutes here because I think it's
Please.
important. So we've been now in business for almost 9 years. We've raised $200 million. We have 500,000 customers in more than 100 countries. And we have about 1 million samples, stool, blood, and saliva samples that we have analyzed metatranscriptomically from all these people, right? And we have enrolled 16,000 clinical research participants over the last 8 years. So an extremely robust scientific platform with metatranscriptomics, machine learning on a very diverse population, and so on. We have a state-of-the-art aging model, biological clock, and machine learned models for many diseases and many phenotypes. So how does all this happen? Where statistically speaking, if you start a biotech company today, you have basically a 95% chance of going out of business in the first 3 to 5 years, right? Statistically speaking. And so people are scared and say, wow, this sucks. The odds are against me. And so it's high risk. But you can de-risk this. And that's what I did. And so how do you de-risk starting a company? Here are the key points. One is understand what problem you're trying to solve and then develop the best tools to actually solve that problem. Okay. And let me give you an example. So what I wanted to do is I wanted to understand root causes of all chronic diseases and cancers. And so going back to our earlier discussion, DNA was never going to provide that solution ever. And so in 2010, I could have said, you know what, microbiome field is getting hot. Let's start a company. We'll do some DNA testing, 16S analysis, and we'll just, boost it up with some PR, and there's a company. But I knew that wouldn't solve the problem. And so that would rely on marketing, that would rely on, other things that are not scientifically sound. And so I said, okay, I know how to solve this problem. All I have to do is work out this metatranscriptomic analysis. And so I spent the 6 years to actually develop it until it was at the right quality level that we can start a company. All other microbiome companies, or the vast majority of them, they're started by people who are business people who say, you know what, microbiome field is hot. What do we need to actually start a company here? And some guy says, oh, we just need to contract a university, core facility that will do 16S cheaply. Another person says, well, we just need a front-end web developer. Another person says, yeah, we just need, some nutritionists to interpret the data. And boom, there's a company, right? And they're like, this is great. It's a hot field. It doesn't take much to start. We don't need our own lab. Let's just do it, right? That is the wrong reason to start a business. You're basically starting a business in order to hopefully hype it up enough to sell it and make money. That's the reason. You have no intention or know-how to actually solve any problems with that business. And I think in modern era, businesses should be used to improve humanity and not just to make money. You can also make money, but why not use the corporate for-profit model to actually solve some of humanity's biggest problems? Because governments are not solving them. Academia, to a large extent, are not solving them. Pharma is not solving them. So we really need entrepreneurs who are creative and who are using the right technology to solve problems. And so de-risking starting a company is, number one, is understand what problem you're trying to solve, and is that problem big enough? If you're trying to solve a problem for 5 people on planet Earth, I wouldn't start it because that's probably not going to work. But if you're trying to solve a problem for 1 billion people, that's a huge problem. Now, do you have the right tools to solve that problem? If you do, then you've just de-risked your company by orders of magnitude. And then the next step is, do you understand what it takes to run a business and start a business and all that stuff? And 20 years ago, if you weren't trained to do that or had experience to do that, you would be like clueless. But today, there are all resources in order for you to understand what mistakes not to make, what to do, when to do it, how to do it. Everything is online. Like, it's free knowledge. Go and learn. So before you actually leave your academic position and start a company, spend 2, 3, 5 years. I spent 10 learning how to start a company and run a company from all the previous experiences, including mistakes. So that's a huge de-risk. Last but not least, have a leadership or foundational team whose number one mission is to solve the problem and not what's in it for me and how much money am I going to make. If you bring one wrong person on the leadership team who is there to make money, you're already dead. You basically already determined your fate, right? So just bring people on who want to solve that big problem, and that's why they're there. And that's what we were able to do at Vio. And so those are the three key things. The rest, literally just on a day-to-day basis, just don't do stupid things. Do things that have been done in the past. It's not rocket science, honestly. So I wanted to just spend some time on that to give people the comfort, because I see so many brilliant academics and they're stuck in a cycle of grant proposals, papers, grant proposals, papers, grant proposals, papers. And if they took off and started a company, they could literally change humanity for the better. And so that's what that was about.
I'm hearing echoes of what Kathleen Carrico wrote about in her Breaking Through autobiography when she kind of transitioned into industry and she saw the different incentives, right? Like, whereas she was in academia, it's publish or perish. And she had to live under these confines of, all right, let's get this freaking publication. I was like, wait a minute, I'm here to do science, Whereas, and then she looked at the funding model, or at least described the funding model that we know well in U.S. academia, where it's not necessarily always in line with scientific breakthroughs that are going to have impacts in the world, and prioritizing that.
Yep.
And I think sometimes, especially in the biology, life sciences, we're not always shown the other side of the fence of, hey, how can we approach this in an entrepreneurial type of spirit? So I appreciate you bringing this up.
Okay.
And again, this is something that Dr. Carrico also speaks on, where she saw the incentives aligned. She saw people who wanted to make a big impact. She believed in the technology so deeply and the ways in which it also in the RNA universe here, that it could impact humanity. And that's what she, just doubled, tripled, quadrupled down on. And then it's had the impact that it's had.
Great. Okay, now let's go to your question, which was, how do we use metatranscriptomic data to make nutritional recommendations?
Yeah. Or actually, maybe even if we stop there, nutritional recommendations is where it's at now. But I think the way you're speaking about it, diagnostics is in the forefront, like way more than just nutritional food dietary recommendations. Or was this always the big mover that you thought was the most important to focus on first?
Well, so that's a big topic that we can open up now. So in terms of diagnostics, today we think disease is inevitable and we need to diagnose the disease. And the main purpose of diagnosing a disease is to give a medication. That's basically the link. Our system today is set up to diagnose a disease and then prescribe a medication. And 100 years from now, we're going to look back at that and say, wow, that was so primitive. We actually waited until people fell apart enough to where they were symptomatic. And then we were giving this drug that had poor efficacy, that had side effects, that just didn't solve the problem, just managed the problem, right? So we're going to look back and say, wow, that was horrible, right? Primitive. So what we need to do is we need to think about the human body in a very different way. So let's go back to my 100,000 reactions, right? If we think of our body as 100,000 fine-tuned reactions, meaning all reactions need to run at a certain rate. If they run too fast, not good. If they run too slowly, not good. And a simple comparison would be cholesterol production, right? If our body produces too much cholesterol, everyone knows that's not good. But it turns out if our body cannot produce enough cholesterol, that's also not good. Cholesterol is essential for our absorption of nutrients, right? And so many hormones are produced from it as well. And so that's one biomarker that is sort of very commonly known. But what if there are 100,000 biomarkers that define a healthy homeostasis? And we have done that ourselves. So we have defined the healthy level of, let's say, 10,000 different reactions, not 100,000, 10,000. So we don't yet know all of them, but 10,000, which is a lot because I'll give you an example that for our biological aging clock, there's only 1,200 reactions, biochemical reactions in the human body that very precisely determine how fast you're aging. So 10,000 is even an order of magnitude larger than that. So it really captures comprehensively a lot of the human biology. So if you take 10,000 biochemical reactions and understand what level of those reactions is healthy, now, if you profile someone at the age of 12, you can immediately point out at the age of 12 which one of those 10,000 reactions are not in the healthy range. And this is decades before any symptoms, literally decades. You can say, these 3 reactions are out of whack, let's fix them. When someone is 25, they're going to have 30 reactions that are out of whack. When someone is 35, they're going to have 300 reactions that are out of whack. And when someone is 50, if they don't do anything about it, right? They're going to have thousands of reactions that are out of whack. And at that time, they're symptomatic because enough things have broken down in their body, right? But what if you can, from birth, digitally profile the human body, and then as a person grows and ages, you identify every single reaction that is not optimal. And if it's too fast, slow it down. If it's too slow, accelerate it so that all 10,000 reactions or 100,000 reactions are all perfectly optimally running. There would be no reason to diagnose type 2 diabetes, right? There would be no reason to do that because there would be no type 2 diabetes. So our diagnostics, what we are now doing, and we have a commercial product with this, we are not diagnosing a disease. We're diagnosing parts of human physiology that are not optimal. And this is years or decades before any symptoms would come on.
Can you speak to that? You just described your team has defined what a healthy microbiome is. What does that mean? Is this something that in the metatranscriptomic world there is a consensus, hey, this gene profile expression is the ideal for a living modern human? Or can you double click on that a little bit there?
Yeah, exactly. So when I say biochemical reactions, I do mean basically metabolic activities of microbes as well as human genes, not just microbes, right? So I basically think roughly today that we are seeing close to a third of these biochemical reactions in saliva, close to a third in stool, and close to a third in blood. It depends on the phenotype. It depends on other things, but we're seeing roughly that many. And so if we're measuring about 10,000 of those, roughly 3,000 per sample type we're seeing. And for those 3,000 reactions, many of those are related to different phenotypes, including healthy phenotype. And so we do have now, so out of 500,000 people that we have in our pool, in our data lake, 550 have that super healthy physiology, where the vast majority and the most of those 10,000 reactions, more than in any other group of people, are running at the perfect rate. They're not too slow, they're not too fast. Does that explain it?
I think I might be missing something a little bit though. So is that to say of the 500,000 that you've tested, what was the percent that you said had that optimal profile?
Well, it depends on the person, right? The more they age, the more reactions are out of whack.
I guess what I'm hearing, especially when you're describing thousands of parameters, to call something that the health, what is good, what constitutes as good? Is
Yeah.
it we've decided this is a good microbiome because X, Y, Z.
Okay,
Yeah.
so let's pick one example. So let's say hydrogen sulfide production by the gut microbiome. Okay, so hydrogen sulfide is this chemical messenger that is very important for the gut health. And so it helps with intestinal permeability, intestinal health, integrity. It helps with gut movement. Peristalsis, I think, is what it's called. And so if you don't have enough hydrogen sulfide produced by your gut microbiome, you have a certain set of phenotypes. If you have too much of it produced, you have a certain different set of phenotypes. But if it's produced at the right amount, you're healthy. You have regular bowel movements, you don't have diarrhea or constipation, you don't have a leaky gut, and your intestinal cells are fed. Okay, so that's basically the level. And so how is hydrogen sulfide produced? It's produced from 3 major chemical reactions. One chemical reaction is production of hydrogen sulfide gas from taurine. And taurine is a very important part of our bile acids. So we produce taurine in the liver and secrete it. And whatever taurine ends up in the colon can be converted to hydrogen sulfide. So that's one major biochemical reaction. The next one is conversion of cysteine and methionine amino acids into hydrogen sulfide. And so if you are optimally feeding yourself with the optimal amount of protein, then all of that protein is going to be degraded and absorbed in the small intestine, and very little will be going into the colon. But if you overeat the protein or you're unable to digest it, then more protein will enter the colon where it's going to be fermented, and eventually it'll be made into many metabolites. One of those is hydrogen sulfide.
This is perfect. That's the exact question I was asking there. Like that pathway you described.
Yeah.
What I'm hearing, especially that biological narrative, has taken countless hours of thousands of scientists understanding what exactly is maybe what would be described as a healthy level of the hydrogen sulfide and maybe the gene expression profiles that could promote for that.
Hold on. No. Other scientists don't have the data to know what the healthy level of hydrogen sulfide is, because we're not measuring hydrogen sulfide gas as a metabolite. We're measuring hydrogen sulfide gas production and consumption pathways. Yes. And so our level, how we define it, has to do with literally the sum CPM of the expression level of certain genes.
Okay.
Right. No one can model those data or have that definition because they don't have these data.
Absolutely. But more so what I was saying, it's fair to say that there have been other scientists who've understood that there's an impact of this gas or these compounds.
Yeah. Oh, yeah, yes. Absolutely.
This is what I care to say. And then you guys bring in the insight, okay, let's look at the proper metabolic pathway, and we understand it. And this is what the crux of the question that I'm having here, though, is that's for one of these of the 100,000 parameters you guys are modifying.
Correct.
Does that body of literature exist for the other, 99,000 different parameters that you're measuring for us to be able to say this is the ideal amount?
It exists for 10,000 out of 100,000. Mm-hmm. And of those 10,000, it's a mixture of domain knowledge and our own data. And the main domain knowledge is the KEGG pathway.
Mm-hmm. Yeah.
That's our curated knowledge. There's some BGCs that are published that are not in KEGG, and there's some domain knowledge that we brought in from our clinical research studies. But KEGG is sort of the foundation, and that's where we started, by the way. KEGG is amazing.
Okay, yeah, and that's exactly what I'm getting at is, what is good? What does it mean? And is the aim then to, if you're looking at someone's microbiome expression profile, that's the good thing definitely throws me off a little bit, because it's almost like it would be unique to the individual, and you'd recommend a diet that, okay, here's your certain profile. This is a diet that would be maybe compatible with your microbiome. But it sounds like what you're saying is rather here's a nutritional profile that would help to modify your transcriptomic profiling downstream.
Yeah, so if you just look at the very simplistic way of looking at chemical reactions, every chemical reaction requires enzymes, which we quantify using metatranscriptomics, and they require a substrate in order to produce a product. Yeah. And substrates are either direct micronutrients from food, or they're metabolic outputs of other parts of the gut microbiome that are used by other parts of the microbiome in a cross-feeding manner. Make sense? So, for example, Faecalibacterium prausnitzii produces butyrate from acetate that's produced by other bacteria such as Bacteroides. They don't. So, Faecalibacterium prausnitzii does its major source of butyrate is acetate and not fiber consumption. So there's many cross-feeding mechanisms. But yeah, so that's a simplified version. But if you just let me explain this example, I'll come back to that.
Please.
So we discussed that a major source of hydrogen sulfide is taurine, or cysteine and methionine from protein sources, or glucosinolates from cruciferous vegetables. Okay? And so there are three pathways that all contribute to the production of hydrogen sulfide. And then there are pathways that consume hydrogen sulfide, and there are pathways that redirect some of the intermediates on the way to hydrogen sulfide into other pathways like methane gas production. Okay, so it's very complex. It's not like three directions independently of anything else, right? It's a very complex metabolism, but KEGG pathways and our own additional data allow us to understand these things. So now we can build a hydrogen sulfide score where positive pathways contribute to hydrogen sulfide, meaning consumption of taurine, cysteine, and methionine, and glucosinolates, and negative pathways mean consumption of hydrogen sulfide and bifurcation of hydrogen sulfide production into methane. So now we build a score where we have a coefficient in front of these pathways, KOs, right? You're familiar with KOs, KEGG orthologs. And now we ask machine learning algorithms, I want you to adjust coefficients such that too much of this is going to cause constipation and too little of this is going to cause diarrhea. So we take from our pool of people and pool of customers, and they have self-reported conditions, or we take but we also take our clinical research participants who we know whether they have diarrhea or constipation based on like the Rome 4 criteria. And now we tell the computer, okay, the ground truth is case 1 is constipation, case 2 is diarrhea, and control is healthy. And now adjust the parameters, the coefficients of all these KOs such that we get the best odds ratio and best prediction. So we train on a certain set. And so let me give you one example. We published a paper in the fall of 2023. We used 10,000 humans for a training set, and we did an independent validation on 50,000 humans in order to report the data. Okay. We did it on 13,000 humans back in 2023. Now we're publishing a paper with 50,000 humans, independent validation. So the independent validation has huge numbers. So it's very trustworthy. And so this is how we build our scores. And this is how we realize what are the optimal levels of hydrogen sulfide for health. Any lower than that, and you will have a certain phenotype. Any higher than that, and you will have a certain other phenotype.
I got about 85% of that. So I think I can land on the understanding here that as you're looking at the transcriptome, there are understood pathways that you're understanding the modulating factors of how these transcriptomes interact with these metabolic pathways. And so if moving on to that then, especially because of the minute parameters we're speaking about, is then the assumption that, okay, certain foods will have substrates that will impact this metabolic pathway in this specific way.
Exactly.
And that's how I can recommend an artichoke over a blueberry because I'm looking at the expression profile you've got.
So the very first layer that the computer is going to look at is what are the pathways that are running too fast? And to slow them down, the computer is going to find foods that contain, or it's not going to yet find foods. It's just going to reach into our food ontology and ask, what are the micronutrients that can act as substrates for the pathways that are running too fast? And the computer creates a list of substrates that we need to eat less of. So it says, eat less of this, less of this, less of this, right? And then it finds pathways that are overexpressed, that are running too fast, that are running too slowly in a person, and say we need to boost these pathways. So we need to provide them with substrates. So it generates a list of substrates that the person needs to consume. And then next step is, okay, let's map these substrates to foods that people buy at the grocery store and say, any food that has substrates to be avoided, avoid. Any food that has substrate that people need to eat, put it on the enjoy list, right? And then it does a lot more fine-tuning beyond that, a lot more. That's sort of the start. There's lots and lots beyond that. We have a medication interaction database. We have weighting of all of the nutrients, because if broccoli has a healthy nutrient and an unhealthy nutrient, what are you going to do with that? There's a conflict. You now have to understand not only the weights of those nutrients, how important is the beneficial one and how important is the detrimental one, but also you have to tie that to any existing phenotype that the person has. You have to tie it to other biochemical reactions that person has, and the ability of you to find other foods that contain the beneficial nutrients so that you can say, do not eat broccoli, or to place beneficial nutrients into the supplement list so that you can tell them not to eat broccoli. So there's just thousands and thousands of logical circuits that all come up with the final version of the recommendations. By the way, in there is included any sort of food sensitivity. that customers self-report. And in there is included also a glycemic response model. So we've built sort of a state-of-the-art glycemic response model, which basically predicts which foods are going to trigger high blood glucose spike and which ones are going to trigger low glucose spike. And so all of these algorithms are mumbled together, and here comes one most optimal solution. It's not the perfect solution. It's just the most optimal for that person, given the limitations of all the combinations.
And the recommended foods is at the resolution of eat some of these, eat some of those, avoid these, and you should be solid. Or does it get to the quantities? I'd imagine
It
quantities,
does
even
get to the
times a
serving
day. Do these
sizes.
things matter too?
Yeah.
Yeah.
Yeah. So we have our recommendation engine puts out groups foods into 4 categories: superfoods, enjoy foods, minimize foods, and avoid foods. So avoid foods, self-explanatory, never eat them. Minimize foods, try to eat them if you can, but small amounts, no more than a serving per day. Enjoy foods, you can eat all you want. You can eat any diversity. This is kind of the largest group for a typical person. You can eat as much of them as you want, but there's nothing critical about them, meaning you don't have to pay any special attention to them. So if you have a list of 200 foods, you can limit yourself to 50 of those, and it's okay. But then you go to superfoods, and that list is anywhere from 3 to 30, depending on the person. You must eat every single one of those foods. Your body requires nutrients that are found in those foods and cannot be found anywhere else. And we were not able to place them in the supplements. And so every category has its own definition. And based on that, today you can just go to ChatGPT and tell it, here's my lists, build me recipes. But we are actually creating that as part of the app. So this year we're going to release the app that'll enable people to create recipes from their own recommendations.
In the 500,000, 600,000 or so profiles you've seen, are you seeing that actually it is very unique to each profile? Or are you also seeing commonalities that maybe mimic the rules of thumb of nutrition that we've known about before of eat whole foods, get some sleep, drink water, make sure you go on a walk every day?
Well, we don't currently make any recommendations that are personalized recommendations for exercise, sleep, or Zen. None of those. We only make nutritional recommendations, and our computers are unaware of any pre-made human diet like ketogenic or Mediterranean. So there is no bias towards any of those. We do adjust macronutrient ratios depending on what their microbiome looks like, what their symptoms are like, and what their goal is, right? So if your goal is to lose weight, or build muscle, or improve mental health, or all kinds of goals, those are going to be taken into account for the macros as well as micronutrients. So there's just a lot of inputs into the recommendations. And no, we have not seen any. Well, I have not spent time looking at 500,000 recommendations to tell you, okay, is there any sort of prepackaged thing? But no, there's no, for example, if you go to many nutritionists today with prediabetes or diabetes, they're going to a large extent, recommend, you lower your carbs. Our computers don't do that. Our computers do not lower carb intake based on the fact that you may be pre-diabetic or diabetic. The only thing our computers do is say, of all the carbohydrate containing foods, here are foods that are going to spike your blood sugar more. Minimize them or avoid them. And here are the foods that are carbohydrate rich that are not going to spike your blood sugar, so eat them because they have healthy ingredients for you. And so the computers are not minimizing any nutrients specifically based on like a condition. It's just personalizing them.
It's interesting that you're not finding those commonalities because you assume there would be, given that, again, on the evolutionary biology side, we all have some similarities into us. And I think I've heard you speak in the past about like blue zones and things like this. And there's the commonalities of, their environment and maybe even their nutrition and maybe even being outside of the urban environments or something like this, where, if you look at their microbiome profile or whatever it might be close to that ideal across these similar types of blue zone areas that, they're not looking at the resolution you're speaking, but they're still seemingly healthy given some commonalities.
Here's my view. My view is if you look at the blue zone on a Greek island and compare it to a blue zone in the mountains of the Philippines, let's say, or Okinawa, okay? The commonality between those two has nothing to do with the microbiome composition or food. It has to do with the metabolic output of the gut microbiome. And there are two completely different combinations of foods and microbiome composition in those two blue zones that still yield the same metabolic output. And the way that has happened is during coevolution, right? So people in Greek islands eat different foods, like they do not have access to sweet potatoes, right? They've never had access to sweet potatoes. And yet they have foods that provide the same metabolic output when combined with their microbiome. If you sequence the gut microbiome of a Greek island resident and Okinawa resident, they're going to be very different taxonomically, like basically zero overlap.
Mm.
Yet, if you look at their functions, biochemical functions, they're going to be very similar. They just require different inputs for different gut microbiome, but they've evolved with those inputs. Does that make sense?
It does. So then I'm curious, then how do you react to, maybe the commonly held assumption that for most people who are trying to just lose weight, it's really a change in eating a little bit less, sticking to whole foods, and even as a nutritional recommendation, do you feel that's maybe in the face of what you're seeing, not going to be something that helps most people?
No, I think that helps most people. That's a good general advice. What doesn't help people is, I mean, weight is not the only parameter that's associated with health, right? There are plenty of people who are not fat or not obese that get a heart attack. There are plenty of people who are not obese who get that type 2 diabetes, Alzheimer's, all kinds of things. So obesity is just one phenotype. We can't just be saying do a caloric restriction, lower your obesity, and you'll be healthy forever. That's not true.
I hear that completely. I see what you're saying. That where people are looking at losing weight, it's a bad proxy. We're looking at one aspect of your health, whereas if we're looking at, if I'm understanding what you're saying, if we're looking at the actual microbiome expression profile, we have way more monitors. We got a lot more barometers actually out here, other than just sticking your thumb up in the air and figuring out where the wind's blowing.
Plenty, and I mean plenty, like hundreds of millions of normal weight people who have mental health issues. That's because their gut microbiome is not producing neurotransmitters, right? And so plenty of people who are constipated and they're skinny, plenty of people who have diarrhea and they're skinny. All of those are abnormalities that have nothing to do with body weight. So just think of body weight as one part of your overall physiology that's visible. But fatty liver disease, you don't see it. Alpha synuclein, you don't see it. Beta amyloid, you don't see it. All those things, you don't see them.
Do you feel, though, in what you've described, is that something most people should be aware of, or are those sort of on the fringes of, the biological outliers?
Which?
You're talking about people who are, seemingly healthy, who are susceptible to, conditions. Yeah.
I would definitely say that there are hundreds of millions of people in the world who are seemingly healthy. And there's a lot who are not healthy, first of all. And second of all, the majority of those have symptoms. And second of all, what's frustrating even more for many of those people is that they go to the doctor and the doctor says, everything looks normal for you.
Yeah.
We don't see any problem with you, right? There are literally tens of millions of people in the world, possibly hundreds, that have some kind of an illness where they go to a doctor and the doctor says everything's fine with you. That's a huge epidemic these days, that the biomarkers that the medical system has measure such crude things and so few things, they can't possibly characterize our entire physiology. We're measuring 10,000 biomarkers and we still don't capture all of the physiology.
It's the exact narrative of whole genome sequence, or rather next gen sequencing in general, right? It's greater resolution. I have more data points, and it sounds like what your team has been able to figure out here is how to translate this technology and RNA-seq expression profiles into giving us the appropriate, not only the data points. This is actually the thing that I've been most interested in this conversation: the context of the data points. That's what matters most. It's not just I know this expression.
I would call it actionability.
Yeah. And how specific that is to different people. And, I think we're getting nearing close to time. So I'm curious if we can end on one other topic here of, I feel like now people have this resolution of, okay, this is the sorts of foods I can eat. These are the ones I can't. It sounds like you want to even increase that resolution a little bit more, make it a recipe, maybe get into the exercise, other types of lifestyle recommendations that would be, in any way promote their health. For a lot of people, though, even those basic recommendations are known of eating whole foods, sleeping, etc. But the impetus to action isn't necessarily, going to be more resolution to the information I'm getting. The blockade to compliance is something I think the medical world, no matter what the tools are, has been the major challenge. So I'm curious how your team thinks about that.
Yeah, so there are two aspects to it. First of all, the vast majority, like 99 point some percent of all nutritional science so far, has asked the wrong question, which is red meat good for all humans or is it bad for all humans? Are eggs bad for all humans or are they good for all humans? And that's the main reason that we have these publications where if you like to eat eggs, you can find trials that say eggs are good for you. If you don't like to eat eggs, you can go find trials that say, oh, eggs are bad for you. And you can make an argument to your friends that eggs are bad for you. The same for coffee, the same for dairy, the same for every kind of food, right? And so when there's so much conflicting information in sciences where scientists cannot agree whether red meat is good for you or bad for you, right? Why would anyone listen? How are you going to convince someone to stop eating red meat if there's clear evidence out there that red meat is very nutritious and very healthy for some people, and that some people who live to 95 healthy have eaten red meat all their lives? Why would someone stop eating something they love if that's the evidence, right? But if we change the paradigm and say, wait a minute, we're not asking if red meat at Viome, we're not asking if red meat is good for all or bad for all. That's not true. We're asking who is red meat good for and who is red meat bad for. And here's the molecular evidence as to why it's bad for you. And it's a publication where we show that odds ratio, you have a higher risk, all that. Now people have much more credible evidence to say, I am not going to benefit from red meat. It doesn't matter that my uncle ate red meat every day and he smoked and he still lived to 95. I am not my uncle. My data tell me that I have to do this. So personalizing it, I think, is the number one step that you have to do. Otherwise, no one's going to listen. So that's one. The second is running randomized control trials. So we are running 4 randomized control trials. We have very good output from 2 of them, and a pretty good outcome from another one from an interim analysis. And so showing in randomized control trials that this actually works, that will help a lot. And last but not least, when we do health economic models and when we show that this works, when you have an employer whose future depends on lowering their healthcare costs because they can't afford it anymore, and when they provide Viome to their employees for free, you're going to have a higher adoption rates. And so it'll be cost free. And then when our app enables people to actually translate all these recommendations into daily living, like what am I going to buy at the groceries? What am I going to cook for dinner? When all of that is enabled, the barrier to compliance is going to be lower and lower. And now you're going to have more and more adoption. And eventually, most people will adopt it. There will still be people who say, I know cheese is bad for me. I know I'm going to die from a heart attack, and that's okay. I want to eat cheese, and that's my choice, and that's okay. No one should be forced to not eat cheese, but all the data will be there for them. So we're making one step at a time. It'll work.
And have you seen any surprises in some of that data where it's like, hey, we assumed this would be the sort of dietary profile that best fits your transcriptomic expression profile, but you know what? I started eating these superfoods. Momo, I'm feeling more malaise than I've ever felt in my life. What's going on here?
Well, we have, I mean, I don't know about that specific example, but let's just make sure that we understand the following. The part of the human physiology that we profile is a part. We're not profiling every single part of human physiology. So if someone has heavy metal exposure, or they have a mycotoxin exposure, or if they have a food sensitivity or food allergy to a certain food, or they have a hormone out of whack, we don't measure those biomarkers, right? So there are many people who have symptoms that are not due to anything that we measure, meaning that if we optimize their system to the max using our approach, they'll still have symptoms because the root cause of those symptoms has nothing to do with microbiome. If they're intoxicated by a mycotoxin, there's nothing that nutrition is going to help them. They have to detoxify their mycotoxins. Same with heavy metals. And so I just want to make sure that we understand that. Second, the limitation of our approach is that we cannot intentionally tune everything. We don't know enough about microbial physiology and human physiology to intentionally tune everything. It's a very complex system. There's so many microbial physiology features that we don't know about, and there are many that are cross-feeding that we don't know about, and there are many that we don't measure. So, for example, 42% of sequencing reads in stool are interpreted functionally. 58% are not interpreted functionally. So there's a ton of dark matter that we don't know what to do with, right? So we need more data. And so think of this platform as iPhone 1. It's awesome. It works. It's great. But it's going to get a hell of a lot better as we learn more and we develop more science.
It's going to be an exciting kind of view of watching your company grow and find its way into the general health system here. I've got definitely now an affinity to try this out, see what my microbiome profile looks like a little bit, and see what kind of foods are recommended. And and it sounds like, right now, Metatranscriptomics is on the edge of even most scientists' understanding of what the heck's happening here. But in the future, you're envisioning it's going to be as common as blood pressure.
It's going to be very, very much a common thing. So when we get to the point where entire national healthcare systems and self-insured employers require this of their employees to at least do the test and see what their recommendations are and offer it for free, it's going to be super widely adopted. And when you can say to someone, out of the 10 different pathways that are making nitric oxide in your body that regulates your blood pressure, 3 are out of whack. Your 7 others are working, which means your blood pressure is still normal, but you're on your way to, having a dysfunctional blood pressure. You better do something about it now before 8 of them are dysfunctional, because at that point in time, you will have high blood pressure. So now is the time to do it. We will not need to measure blood pressure anymore, because you will prevent blood pressure decades before it goes out of whack, right? So the whole paradigm about the human body changes. And I know you want this to be a scientific sort of a podcast, but I want to make a metaphor to airliners. And so before we implemented preventive maintenance for airliners, they took off because the engines ran and all the controls moved. But in flight, something broke and the whole plane crashed and all the people died, right? And that's exactly what we're doing to our bodies now. We're waiting until something breaks and we're sick. And now we're trying to fix it. Trying to fix an Alzheimer's brain is not simple, because so much pathology has already taken place, right? So what we implemented for aviation back in the 50s and 60s is a preventive maintenance program. Before that plane takes off, why don't we go and open up the panels and inspect all the critical parts that are needed? And if a part is worn, let's replace it before it breaks in flight, right? And so the example I like to give is that this Airbus A380, which is the most complex airplane ever made, it has flown 1 million flights without a single fatality. And it's the most complex plane ever made. 1 million flights with no fatality. That tells you that the preventive maintenance program is working perfectly. They don't need to measure how many wings have broken in that plane, because it's never happened. They don't need to measure how many rudders have broken in that plane, which would be sort of the metaphor of an illness. They identified that a jack screw in the rudder assembly has worn out after so many flights. They replace it, and they know that that rudder is going to work every single flight. And that's what we are bringing to human body. Exactly. Preventive maintenance, right? Let's identify parts that are malfunctioning before they completely break, before your brain completely breaks and you're unable to even know who you are. Why don't we intervene decades before that, so that you don't even need to have a test for Alzheimer's, right? Today, we don't screen people for bubonic plague, right? You're in the infectious disease area, right? I compare today's status of chronic diseases and cancers to the dark ages in the 13th century, where when a bubonic plague came, you either died, whoops, bad luck, or you survived, good luck, That's it. We knew nothing about what was the root cause, how it was spread. We had no way to treat it. Today, we're not testing for bubonic plague every single day, and nobody wakes up today saying, shit, am I going to die of bubonic plague today? Nobody does that because we understand everything about the root cause of that disease, how it's spread, how to treat it, and so we don't need to worry about it. Why can't we have the same for multiple sclerosis? Why can't we have the same for Alzheimer's? Why can't we have the same for pancreatic cancer? We can. We have all the science and technology. Someone just has to do it.
You bring up a really interesting point. This actually maybe ties back, if you got a little bit of time here, to the conversation of what is good. And I think that analogy you brought with the Boeing Airbus there, or the Airbus flight complex, million flights, whatever, nothing going down, that was based on open communication and standards of what is the standard. The FFA regulating that and defining what are the parameters that make up what is good. I think, outside looking in, that's what I'm trying to get at. How do we know Viome's approach to establishing what is good? And if I got another metatranscriptomic test at another laboratory, how can I compare and make that consensus so we all have a shared agreement of what is good? How did we define what is good and all the rest? I'm curious if you have any thoughts to that.
Well, I mean, if another metatranscriptomic company pops up, they'll have to go through the same thing that we've gone over the last 8 years, which is analyze a ton of data and then compare them to different phenotypes and establish what are the healthy level of biochemical activities for all these thousands of different biomarkers, right? So they'll have to do that. We're not going to tell them how to do it because we're a business, first of all, and second of all, their method may be different. There are many different metatranscriptomic methods with different biases. We may have a systematic bias, they may have a systematic bias, and both of us can achieve great success, but with different coefficients associated with those different biochemical reactions, right? So they'll have to do that. Yeah, that's it. Pretty simple.
And I'm only commenting that as the general perspective of, how does this become more standardized and things like that. It just speaks to the complexity of it. If everyone's going to be testing the metatranscriptomic differently, how do we have that standard of, what is considered the healthy profile? But, that's where my conversational or my brain goes to in this. It's hard to,
we haven't gotten there. There's not been another metatranscriptomic company yet, so we don't have to worry about it. And
fair.
I would say, it's been a 15-year journey to get to where we are today, and we still have a lot more work to do.
That's awesome.
And at some point in time, nutrition will be viewed as medicine. Today, the only way to get to medicine is via the FDA approvals. And the FDA approvals are very much geared and focused on drugs, meaning single chemical entity that does something. Nutrition is far more complex than that. So you cannot get like a diet FDA approved. That doesn't make any sense, right? And so is there going to be a completely new framework, like a completely new agency that basically has a way to run randomized controlled trials on foods where you're actually, so what I would like is for us to be able to say, okay, we've locked down our metatranscriptomic method. It is what it is, whatever bias it has. We've locked down our mathematical algorithms. It is what it is, whatever they know. If we combine the two and then people are given the food that is output from that in a randomized control trial relative to some normal diet, does this actually improve some condition? And if we show that in a randomized control trial that it does, then we should be able to make a health claim. Today, that's not possible. There's no regulatory infrastructure framework for that. Whether it's ever going to be like that, I don't know, but we are moving forward in the wellness space without making any health claims, and it's fine. I think it's fine.
Momo, thank you for the time. A fantastic discussion. I hope we run into each other here in Washington State. And we'll see everyone in the next one.
Okay, thank you for doing what you're doing. Good day.